Hi guys, and welcome to Grade 12 Life Science. Finally, I know that you guys have been chomping at the bit for your turn, and finally it has arrived. We are going over such an exciting section today. It's genetics, guys. It's all about us. It's about what makes us tick and what makes us happen. We've got Aslam with us, and I know that he's dying to start the lesson, but you all know that the beginning of the show is all mine. So guys, what I need you to do, this is very, very important. This link, this is the link that you need to know. Please write it down. Please visit it. I cannot stress it enough. It is learnextra.co.za/live. In that link, you're going to get our schedules. You're going to be able to get the notes, including the notes for this lesson, so you can download them. You can work along with us. I will post the notes now as well, little fast link. And you can get all of last week's lessons as well, which have been uploaded to YouTube. So guys, this is the link to go to throughout the year. This show is also proudly sponsored by Macmillan. Thanks guys so much in this awesome Solutions for All book. So some of the stuff we're going over, the content comes from here. You guys rock. So I'm not going to waste any more time. Chat to me on facebook.com/learnextra, also on Twitter @learnextra. I'm Indy and I'm here to be taking all of your questions, and I will pass them on to Aslam. So Aslam, you've said we're having a fun lesson today. Let's take it away.
Thank you, Indy. Welcome, Mindsetters. I'm hoping you had a long week rest, and now we're back to move on with the chapters. Indy's mentioned something that genetics is exciting because it's about us. Guys, where was Indy all these weeks? Everything we've been doing is about us. DNA was about us. Protein synthesis was about us. Meiosis was about us.
But Aslam, that's why I love life science so much. It's all about us and how we work.
I
It's
asked
so
my kids
interesting.
this morning, I said, isn't this your most exciting lesson? And some were hesitant, and they're scared to say no. And then I said, well, we're going to be doing sexual reproduction.
And then they
Can
got,
you do that in
then
English?
they got excited.
They got excited. They got
Teenagers.
excited. They got excited. Guys, we are busy with a section on genetics, as Indy has indicated. I like that.
Indicated.
Vindicated. And we are going to try to make this lesson as fun as is possible. I have learned whatever I have learned from a man, Mr. M. Baba, and today I am dedicating this to him. Mr. Mahmood Baba was my biology teacher, Indy, and he was full of life.
It's amazing.
And that's what inspired me to be there today and where I am today. So, Mr. Baba, if you are watching, if this lesson is exciting, it's because of you. And if it's not exciting, it's still because of him.
And all the mindsetters thank Mr. Baba for making you so amazing.
There we
Aslam.
go. There we go.
Yeah.
Right. What are we looking at today, guys? Very simple. We are looking at the definition, obviously, of genetics. We are looking at Mendel's concept of dominance and the law of segregation. We also want to cover the terminology that deals with genetics. Remember, we've been stressing this point all the time. Terminology. Section A in your paper is 50 marks. Most of these 50 marks are dedicated to terminology. So if you do well in a terminology, you're on your way for success. You cannot fail the subject. And those that are getting an A, remember, you can lose your A in Section A and in Section C. If you misunderstand the essay or you mess up with the terminology, you can lose your A. So you've got to be on the ball here. We also then want to look at the main feature of this whole thing of genetics. Why do we learn all 1, 2, 3? We learn 1, 2, 3 so that we can solve genetic problems. In other words, we want to know how, when we take a red flower with a white flower, what is going to be the outcomes from there. Or a blue-eyed person and a brown-eyed person, what are the probabilities of their children being blue-eyed or brown-eyed? Indeed, exciting stuff. Mm-hmm. We're going to look at all types of problems: complete dominance, incomplete, codominance, and we're going to look at the blood groups. By the way, coincidentally, again, a lot of puns today, coincidentally, blood groups is an example of codominance as well. So that now that we've outlined what we want to do, let's get straight into it. We're not wasting time today. First question was the definition. So what is genetics? Obviously, genetics is the study of inheritance of characteristics. In other words, genetics, the study of genetics, wants to find out how characteristics are passed on from parents to offspring. This interesting concept. So we will look at that as we go along. What are these characteristics that we are talking about. Let's look at humans, for example. Just a few interesting characteristics. For a start, let's look at the hair. We get people that are blonde hair, we get people that have brown hair, black hair, etc. We get people with straight hair, we get people with wavy hair. We get people with brown eyes, blue eyes, green eyes, and these days you can wear orange and other colors as well if you put the right context on before you start the day.
But that's not genetics.
That's not genetics. That people will be called an acquired characteristic, not an inherited characteristic. Keep that in mind, Indy, because we're going to be talking about acquired and inherited later.
I will.
Sometime.
I'll keep it up
Keep
here.
that there, right? Straight nose, upturned nose. Well, upturned nose, then, Indy, is not always when you're proud. Okay. All right, a projected chin or a receding chin, as you can see there. You get the lobed ear there, or an unlobed ear. Also, they talk about attached ears and unattached ears. So if you have a lobed structure, that means it's an unattached ear. If you do not have this lobe, your ears are attached. That means they directly there. You don't have a free little pinna to play around with.
I bet you all of the mindsetters are touching the ears. If you're touching your ear now, post on our Facebook page and let us know.
Immediately. Good. There we go. Those are some of the characteristics. Now we go straight to the terminology. The first one is an allele. And before we go into what an allele is, we can go back to meiosis. And remember, we spoke about homologous chromosomes. There we have a pair of homologous chromosomes lying next to each other. And on this homologous chromosome, there may be a gene in this area here. The gene that's in a homologue, or the homologue, the partner, at the same place, the same address, the locus. The locus is the point at which the gene is situated on a chromosome. So the one that is in the same locus as the partner is known as the allele of the other. So we get a pair of alleles because there are always two genes that control every single characteristic in your body. One gene you receive from your mother and one you receive from your father. One of maternal, one of paternal origin. So these two genes are controlling the particular characteristics. So the one is the allele of the other one, and vice versa. These alleles can be identical. Now remember, there's a big difference from coding for the same characteristic and being identical. Coding for the same characteristic simply means it either coding for eye color, size of nose, size of bum, and the rest of it. But identical would mean that if it is coding for blue eyes, if we're talking of eye color, then the other one must also code for blue. So if they're both coding for blue, then they are identical in that. And we're going to come to that terminology as well just now. Let's look at how this plays itself out. There we go. In terms of this particular one, we have a genotype that is capital B, and a phenotype that says brown eyes. Again, two new words again, phenotype and genotype. Genotype, the core word is gene. Genotype, so that means what's inside. How are the genes coding? Are they identical or are they different? If they're identical, they would be homozygous. And if they are different, they would be heterozygous. So if this was, in this case here, it's two capital B, so they're identical, so it is homozygous. And if it was capital B and a small b, it would be heterozygous. And as we see in the example here, second combination, there you have it. And the phenotype, notice next to phenotype across the board there, we're seeing brown eyes or blue eyes. So the phenotype is not talking about whether you're homozygous or heterozygous. For phenotype, for features. So the phenotype talks about how this gene is expressed outside. How do we perceive it? So if it is capital B, small b, although it's heterozygous, we can't see whether you're heterozygous or homozygous. But we can see whether you have brown eyes or blue eyes. That brown eyes or blue eyes is the phenotype, the feature that we are talking about. Remember, guys, I'm telling you, in this section, the terminology is boss. If you can understand the terminology, the rest of the lesson is easy. So there we have covered homozygous and heterozygous. Homozygous would mean when the gene and its allele are identical for a particular characteristic, we say it is homozygous. For that characteristic. Remember, you can't talk of a human being or a plant as homozygous. You talk of a particular characteristic. Because in one human being, I may be homozygous for attached or unattached ear, but I may be heterozygous for brown eyes, etc., etc., etc. So when we're talking about these things, we're talking of a particular gene and its allele. Heterozygous is when the gene and its allele are different for a particular characteristic. Genotype and phenotype, we have explained. Genotype refers to the genetic makeup of the organism. And the answer of a genotype will always be either homozygous or heterozygous. So genotype talks about whether the organism is heterozygous or homozygous for a particular feature. And phenotype, for features. So it's the outward appearance of the particular characteristic that we are talking about. In other words, how does this characteristic manifest itself outwardly? Dominant and recessive, these are not very complicated words. These are English words. Dominant, to dominate, to be the stronger one. So the dominant gene in genetics is the one that is dominant over the other one. So when it is there, the other one will not show. Now let's go back to our example. Look at this particular case. Both the gene coding for brown eyes, capital B, and the gene coding for blue eyes, small b, are both in this individual. But in the eye that you see there, the eyes are brown. So when the dominant one is there, the recessive one will not show up. I call this, when the cat is there, the mice won't play. So in other words, when the cat is away, the mice will play. What am I saying, guys? When the strong one, the cat, is there, we can't see the mice. There will not be anywhere to be found. However, when the cat is away, there's no capital B there. So the cat is gone. So when the cat is away, the mice will play. Now the person will have blue eyes. So the recessive characteristics, boys and girls, ladies and gentlemen, whoever is watching, the recessive characteristic can only show itself phenotypically in the homozygous recessive form. It must be two small letters. These are important factors that we need to learn. Okay? Some dominant and recessive characteristics in our body. The eye color, brown is dominant, blue is recessive. Hair color, dark hair dominant, blonde hair recessive. Hairline, widow's peak, dominant, smooth hairline, recessive, and so on. You can look at this in the book as well. It is there. You can have a look at it. Remember, dominant does not mean, in the strict sense, dominant. Some may say dominant, that means it's the most popular one. No. Dominant means when that gene is there, that's a strong one, the other one will not show. It doesn't mean that the dominant one will be the most popular one. In some cases, the recessive one can be also more popular than the dominant one. By the way, deafness is caused by a dominant gene. Now, you look around you, there are less deaf people than people that can hear. So obviously that tells you that the dominant one is not always the one that is more popular or more frequent in the population. Now, what I have with me here today, Indy, is some balloons.
I'm excited.
We're going to play with them quite a bit today, but I'm going to start with just a few. Let's take some yellow ones and some red ones. Now, remember we said homozygous. Homozygous is when the gene and its allele are identical. So, if I ask you for the genotype of this, you'll tell me homozygous. If I ask you for the phenotype, what can you see? What color? Indy?
The red one.
Red. So,
red.
We are seeing red. So, genotype of this particular organism is homozygous. The phenotype is red. Take that one away. If I put that one up, genotype again, homozygous. And phenotype, now what can you see, Indy?
Yellow.
You sure you're Indy?
I'm sure.
I'm hoping you're not colorblind. Okay, so with that, guys, just think about what I'm saying here now. Think about what we said before. When we come back, we're going to start playing with these balloons. With that, I hand you back to Indy.
There's a break time already.
Yep.
I can't believe it. Okay, guys, before we go to break, this one is actually for the grade 11s. Knowledge is knowing that a tomato is a fruit. Wisdom is knowing not to put it in a fruit salad. We'll see you now after the break. Hi guys, and welcome back. I hope you're having as much fun as I am. I'm just having a look. A gentle Floyd said, "Lol, he was touching his earlobe." I know, I couldn't help it. See, there you guys are doing it again. Toshido was doing it, and Tabiso. Tabiso says, "Lol, I'm touching my earlobe now." And Refiltlwe said, "Me too." So guys, it's all about, hey, it's subliminal messaging. Okay, Aslam, take it away. We're having such an awesome lesson, but I do need to say to the mindsetters, please post your questions on the Facebook page, facebook.com/learn extra. You know what? Aslam and I love taking life science because we like to chat, we like to talk about stuff, and we'd like to help you guys. Guys, there's only a silly question. The only silly question is the one is asked after the exams, because then it's too late. This is a community of mindsetters, and we are all helping each other out. Right, Aslam?
That's for sure. We need those questions on. That's a welcome back, guys. I'm hoping you enjoyed your break. We're moving on with what we were talking about earlier. All this knowledge that we are developing in the section started off with a guy called Gregor Mendel, who, as we say there, is known as the father of genetics because he actually laid the foundation for whatever we've learned. Remember, Mendel made or rather made these discoveries or these theories before we knew what meiosis is, before we knew what DNA or the gene is. So he didn't explain it in terms of genes. We're explaining his theory today using our knowledge of meiosis and genes as well, and DNA. He was basically an Austrian monk. He experimented a lot with pea plants, which were growing in the monastery where he was working. And he liked to use these plants because they had clearly discernible characteristics, meaning they were either or. There was nothing in between. And we'll talk more about that just now. He understood that there was something that carried traits from one generation to the next. He called it a factor. What do we call it today? We call it a gene. We're saying that the gene is passed on from parents to the offspring, and this gene carries the characteristics. Mendel said that there were two factors that controlled every characteristic. Just now I told you there are two genes, and these genes are called alleles of each other. If they're identical, homozygous. If they're not, heterozygous. He worked with more than 20,000 plants. Why do scientists use such big samples? Why do they repeat experiment? And this, guys, touches on what we call in life science, or in science anyway, the idea of reliability. The more you repeat the experiment and you're getting the same result, that tells you that your results were reliable. Remember, reliability deals with the actual repeating and getting more samples, etc. Whereas validity talks about the method of an experiment. If the method is flawed, the validity is affected. The reliability means if we do this experiment again, would we get the same result? Why did he use pea plants? Firstly, they were easy to cultivate, easy growing, they grow very fast. Lots of progeny. They gave lots of offspring, so it's nice to work lots of numbers. You can see what is happening there. They have pure breeding strains that are available. In other words, pure breeding strains is the same as saying homozygous. When we talk of a pure breeding strain, we mean that the gene and its allele are identical. That means it can't make anything else. It can, if it's purple, it's only going to make purple. If it's white, it's only going to make white, etc. They showed obvious differences in the traits that I told you earlier. Easy distinguishable. It's red or white, round or wrinkled, and so on. The parentage can be controlled. I almost said percentage there. Parentage can be controlled. Why? Because peas are self-fertilizing and can be crossed artificially. In other words, how can we control it? We can cover the self-fertilizing plant, and we can go there and brush off pollen from another flower onto the stigma there. So this is why he used pea plants. What were the characteristics he studied most? He looked at seed color, yellow or green. Seed shape, round or wrinkled. Seed coat, gray or white. Pod color, yellow or green. Pod shape, inflated or constricted. flower position, axial or terminal? Are they on the sides or are they the tip, the terminal tip there? The stem length, are they tall or short? Can you see? It's either or, nothing in between. Tall, short, we didn't talk about medium. Tall or short, nothing in between. Let's look at one monohybrid cross that he did. He took a tall, pure breeding plant and he crossed it with a short, pure breeding plant. What happened in his experiment is that in the first generation, all the plants were tall. So what we're showing here? The law of dominance. Why? Because the tallness is dominant over the shortness, so you can't see the shortness here. When we take these offspring and we cross them, he found that 3/4 of the seeds grew into tall plants and 1/4 grew into short plants. So 75% were tall, 25% were short. So in other words, something happened here. Something that disappeared. We had tall and short. We put them together. We only got tall. We took these tall guys or these tall plants and we mated them again, and we find that in 25% of the offspring, they were short. That recessive characteristic came back. So the law of dominance sort of went away, but it's not that it went away. We will see how that happened when we look at how this problem works. Now, before we go there, let's just do a simple thing that we started with. Remember I said when we started, when we have these two balloons together, the red ones, that means they're coding for red. They are homozygous, that's genotype, and the phenotype is that they are red. If I take them now separately, a yellow and a red, I change it around now. Red is dominant, Indy. Red is dominant. So genotype would be heterozygous, and phenotype?
Zygous something.
Phenotype would be red, Indy.
Oh, red and yellow, okay.
For features, Indy, you've got to get with the program here. Otherwise, we're going to send you out.
You know what? I'm so busy Facebooking here.
Okay.
I'm concentrating now. I'm concentrating now.
So, guys, remember, this is heterozygous. If I drop that, that's still heterozygous. If I drop that, that is homozygous, and the phenotype would be red.
Red.
Right?
Yes.
If I pick that one up now, it is still homozygous, and the phenotype?
Yellow.
Yellow. But if I drop that now, again, now it becomes heterozygous.
And red and yellow.
Red, red.
Because red is the dominant one.
There you go. Red is the dominant one. If we change the story now and I tell you yellow is dominant, Indy?
Red.
Yellow is dominant.
Oh, no. But you told me red was dominant.
I told her red was dominant earlier. I changed it to yellow. Can you see how you can play around with this in your own space? You can use the same thing we're talking about in your own space, and you can get ahead with it. You do not need a textbook for that type of stuff. You'll use your textbook as your guide. Play with your jelly tots for all I care. Get the jelly tots, play them around, move them around. And what I'm going to show you now is how this whole thing worked. Now, if we take red as being tall, okay? If we take red as being tall now, and we say that a red parent, or a tall parent, sorry, and was mated with a short parent. So each parent, each individual has two factors, Mendel said. We say it has two genes. So there's your two parents, and we put them down there. Obviously, when meiosis takes place, the gametes separate. All we do is we separate the gametes. They move apart. Because in each sperm or egg, there'll only be one factor. This is known as the law of segregation, that when meiosis takes place, Mendel didn't know this. He just said that when the gametes are formed, the two factors separate. And that's what we're having there. And the yellow ones will form yellow gametes. If we have to put them together, we're going to get all that are going to be heterozygous, and indeed, if red was or tall was dominant, tall now, we're not talking of color now, right? And red is tall, yellow is short. When they together, it will be?
You'll be medium, it'll be red.
No.
Is dominant. Yellow.
Tall is dominant. When the cat is away, the mice won't play. So it'll be tall.
Tall,
Right.
exactly. You'll be
Oh,
tall. You
oh.
won't. You won't. What about ending
There's
up
no medium.
Not
Remember,
medium
but
height.
I'm
Hey,
using
like a
this
tall
in
and a
pea
short,
plant.
and then it's
In humans, indeed, it will change. In humans, we have what we call polygenes. That means many genes control height. That is why we get a short human, we get a slightly taller. We don't have just short and tall.
tall.
So that's multiple genes, actually. Sorry.
Okay, so I must just
Multiple
think
genes.
plants.
We'll
We're
talk about that
we're
later.
talking
Right
about
now
the
we're
pea
talking about
plant here, guys.
Okay, so we got that situation. They're saying now we take these parents and we make them. What's going to happen? 75% of them will be, I'm going to try something serious here. I can't get it right, but you got 2 of them that will be homozygous, that will be tall, like this. You will get others that will be heterozygous, still be tall, and then you will get homozygous, short.
Short.
So this is your 25% that come into the play when we are talking about this. I'm hoping we got the picture in terms of that. Now, we don't always show that way. We have to show it in a scientific way. And here's the way you're going to do it in your diagrams. And there we have it. Notice the setup, the layout is absolutely crucial here. Parental generation, we have tall and short. The phenotype, there's the phenotype is giving you that. The genotype, capital T, or small t for short. When we separate them, remember here, because the gametes will all be the same, we only have to show one here. We can show both, and there will be T space T. Just leave a space to show that they are gametes and they have separated, so you are displaying Mendel's law of segregation. So your capital T's will be separate and your small t's from that parent there. When we put them all together, this is what's going to happen. If we take this line, go down, and that line together, they give us a small big T and a small t, heterozygous, but because the cat is around, they will all be tall. Now we take that lot, the tall ones that were heterozygous. Again, phenotype, genotype, meiosis takes place there. There's the gametes. Notice in this one, because the gametes are different, they're not both capital T's or both small t's. We show them separately here. So there's your one gamete, there's the other gamete, there's another one, and there's another one. And when you join them with a line diagram like this, this is your outcome. Tall, 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 short. 75% were tall, 25% short. Also, we can say it has a ratio of 1:2:1. One homozygous dominant is to 2 heterozygous is to 1 homozygous recessive. That's what we're getting. We prefer you to use Punnett squares. Punnett squares are neater, and the lines are not going all over the show that you can get confused. So what is a Punnett square, you ask? Let's show you that. There you have it. So what we are doing in a Punnett square, we're showing the gametes on the left-hand side and on the top. And in that, we have the Punnett square itself. So what we're saying is, with that T and that T, what do you get? Capital T, capital T. With this T and that T, you get capital T and small t. With this T and the big one, you're going to get capital T, small. By the way, because it's a multiplication, it's a law of probability. Whether you write small t, big T, or you write big T, small t, it doesn't make a difference. 5 times 4, 20. 4 times 5, still 20. Okay? And the last one, this small t and that small t, gives you this 25% that we are talking about. Notice how they say it here. Genotypic ratio, 1:2:1. One homozygous dominant, 2 heterozygous, 1 homozygous recessive. And phenotypic ratio, 3 tall, 1 short, or 75%, 25%, depending what the exam asks you to do that. So this is what a Punnett square looks like. What we did to the balloons was to get away from the Punnett square with letters, and we're using the actual balloons to put them together in what they are doing. Oops. All right. We move on. By carrying out these experiments, what Mendel did with the plants, he determined that 2 alleles for each character segregate during gamete formation. He didn't know this that this was meiosis that caused this. Anaphase, when they separated, they moved apart. And this called the law of segregation. I think at that point, it's in between a definition. The definition is done. Whatever follows, we'll explain after the break.
Fantastic. Guys, what I'm actually going to do is I'm going to post that definition on the page as well, so you can put it on your phone, you can download it, so you've always got it to use as a reference. Also, remember to help each other out on the page, guys. It's facebook.com/learn extra. Help your friends out. If you see something on the page that you understand, please help them, because sometimes it's really fun to learn by helping others, and sometimes that's also an easy way to learn. So what we're going to do now is we're going to take a quick break, get your energy up and down. That's why I literally just jump up and down during the break, and then Aslam and I will go over the questions on the page, and we'll be answering them when we get back from the break, which is now. See you just now. Hi guys, and welcome back to the show. We really are having such a fun time. Lebo Khang said here on the page, says, "Hey Mindsetters, I'm in grade 9 and it's so good for me to watch these shows because I'm really into science and I love it." So would you say it's a good thing? You know what, Lebo Khang, I would say that it is a brilliant thing, especially life science. It's all about the study of you and the universe and how everything works, how the world works. It's amazing. So keep tuning in. What do you say, Asma? Even if you're in grade 9, still okay?
Lebo is my star for the day.
Fantastic.
Anybody that loves life science.
You've got another star for the day, though, as well.
Yes,
and it's got to do with a question.
Yes.
Am I allowed to ask it now?
Please.
Okay. So now this question comes from Lebs Lamini, and she says, "What is a monohybrid cross?" And then this is where the hero comes into the picture, where Gentle Floyd.
Gentle Floyd is our hero as well, because he answered that question perfectly, and he went further to explain even further lots of that. Remember, monohybrid cross is what we are doing at the moment. Mono, one. Hybrid cross, taking one set of characteristics at a time and crossing and looking at the probability. Fortunately for you, you don't do the second one, which is called dihybrid. That means we take two characteristics. Imagine taking a tall plant that has round seeds with a short one with wrinkled seeds. When we do that, obviously we're looking at two characteristics at the same time. It's called dihybrid. You don't have to worry about that. It's out of your syllabus. We look at monohybrid. So the example we looked at above level was the monohybrid cross. We looked at one characteristic at a time. What we did with the balloons was also the same thing. We are looking at one characteristic at the same time. Modern interpretation of the law of segregation says, remember, Mendel said the two factors segregate. What do we say now after we've learned? The characteristics of an organism are controlled by a pair of alleles. We already spoke about this in DNA, that for every characteristic, there is a pair of genes. These pair of genes are called alleles. Okay? And these alleles separate into different gametes as a result of meiosis. Remember, anaphase, the chromosomes move to opposite poles. Anaphase 2, the chromatids move to opposite poles. So when they became gametes, so the two factors or the two genes were carried on these chromosomes, and they were separated so that each sex cell or sperm or egg carries only one of the two. So they have separated. So the law of segregation states that for every characteristic, there are two genes or a pair of alleles. These alleles separate during meiosis. As simple as that. Law of segregation. We're still talking about monohybrid, and the one that we have discussed before of the tall and the short. Obviously, one was dominant, one was recessive, and this shows complete dominance, when one completely dominates the other. So when two individuals with pure breeding contrasting characteristics, contrasting means one is dominant and one is strong in this case. The F1 generation all display the dominant characteristic. So this is complete dominance. Because why I did that now? Because the next one we are talking about, not all the characteristics follow in this pattern. Some show incomplete without the pause now. Incomplete, not incomplete. This is incomplete dominance. Now here what happens? Neither allele is completely dominant over the other. For example, if we take a red balloon, if we take a red, well, let's call it a red flower in this case, and we take this red flower and we cross it with a yellow flower, and the resultant offspring are all orange. So what has happened? Neither of the two characteristics were dominant over each other, and therefore none of them show in the offspring, in the immediate offspring. We get an intermediate color. So red and yellow gave us orange. In nature, the example that's on the board, very simple. We're taking snapdragons as flowers. White flowers and red flowers give us the pink variety there. So neither one of the two is dominant over the other. So they're not dominant at all. All right? We go one step further. I'm going to go back to how we're going to work it out just now. Others display codominance. So we had complete dominance, we had incomplete dominance, now we are talking about codominance, where genes have alleles that are both expressed in the heterozygote individuals. An example, you see this in a book, you have your reddish cow with the white cow, and you get roan. Roan is a color which has reddish hair and white patches on it. So that means both the red and the white show up in the offspring. Now, in our example of our red and yellow flowers, if we mate them and we get this following result,
Look at that.
so we have a yellow flower with red spots. Or we could have had a red flower with yellow spots. So both characteristics are showing up in the offspring. So in this case, we are saying that this is codominant. They're sharing the dominance. Are we clear with this, guys? You can't forget this now because you're seeing the color in front of you. Now, let's go back to our incomplete dominance. We gave the example. Now we want to say, how does it work? So we said that the father, not the father here, the male flower and the female flower, if the male flower was red and the female flower was white, so we have given the phenotype. Notice I am showing you here as well the format of how to answer this question. The genotype, if we're saying we're starting with homozygous, so that will be capital R. And notice, normally, we would have shown small r because of our complete dominance. We move away from this because they're neither dominant over the other, so we use capital W's. You have to use capital letters to show that there is no dominance between these two. So there's your genotype coming out there, and meiosis takes place, and the gametes would be R, R, W, W, separated. All we're saying is that meiosis separates these guys. And when we use a Punnett square, we write the word fertilization here. Here we will put R, R, W, W, R, W, R, W, and so on. They're all going to be RW. And therefore, our individual will be 100% hetero and 100% pink. They'll all be pink. If we take the pink variety and we mate them, then we will get the ratio of 1:2:1 in the phenotype generation as well. We get a 1:2:1 and 1:2:1. 1:2:1 genotype, homozygous, heterozygous, homozygous, and red, pink, white. That's what we will get in our result of this as well. Now, in the next one, we also ask you to do the same thing here. So this was red and white. Again, we can use the same letters, R, R, W, W. I'm not going to do the gametes because you've already learned how to do that. So we have the same thing here, but look at what happens because this is codominance. The genotype remains the same. They will all be RW. Heterozygous. But the phenotype would be this color called roan, or we would say red flowers with white spots, if we're talking about flowers. Or if we take cows that are white and black, we get white cows with black spots, or black spots with white cows. Black cows. Black cows with white spots. Indy, you're not helping me here.
I'm watching you. I find it very funny.
Okay. Notice, guys, look at the
feel?
mark allocation. Look at the mark allocation. You're getting a mark for giving the phenotype. I mean, the phenotype there. Sorry, there we should have actually. Sorry. Good that we went back there. We wrote the wrong thing in the wrong spot there. Good. Lucky we went there. Guys, phenotype. It's red and white. Remember, phenotype refers to the for pheno features. And here then we would say genotype would be there. So watch what you're doing. You're getting a mark if this whole sentence is, well, you can't call it a sentence, but this whole line is correct. And you're getting a mark if this whole line is correct. So you're getting one there, one there. You're getting, if your gametes are all correct, R, W, you get another mark there. For the Punnett square, you get another mark there. And watch what they say at the bottom. For writing P1 and F1, or parents and offspring, you get a mark. And for writing meiosis and fertilization, both these things must be written in the correct position. If it's not in the right place, obviously, if I can just go and write meiosis and fertilization anyway, they're not going to get marks for that. You've got to put it where it happens. And where does it happen? In the formation of the gametes, meiosis will come here. And fertilization will come after the gametes have been formed, in line with the Punnett square. Keep this in mind and follow for every genetic problem. Follow this pattern. Otherwise, you will lose marks unnecessarily. Any questions? Any interesting questions, Indy?
Yes, there's a very, very interesting post here. It's from Kamogelo. It's a very serious one, Aslam. She says, I'm a young scientist and I did my test and I found out that a fact that Aslam is awesome. That is what it says on the page.
She's got a hypothesis. That's not a fact. It's a hypothesis.
Hypothesis.
Yes. We'll have to test that hypothesis. Kamogelo, we need to test that hypothesis. Okay.
Let's test it. We're testing you now.
Right. We move on to multiple alleles, guys. So we look at complete, incomplete, and codominance. Now we look at multiple alleles. When we said that something is either brown or white or brown or black or whatever. Now we're saying that sometimes there's more than two alleles that control a particular characteristic. But in multiple alleles, generally, only two of these alleles will be found in one individual. Although there's more than two, but only two will be found in one individual, in multiple alleles. Okay? And the three types of blood groups we have, or rather, the alleles are A, B, and O. Not the types of blood, but the alleles that control our blood groups are A, B, or O. I told you each one has only two. Blood types follow codominant and simple dominant inheritance. It shows both. It could be codominant or it could be complete dominance, simple complete dominance. Now, the A and B alleles are codominant with each other. So A is codominant to B, B is codominant to A. The A allele and B allele are completely dominant over O. So both A and B are stronger than O. Now, how do we show this? This is important. The way we worked out the tall and the short earlier, the way we worked out red and white, the format stays the same, but the symbols that we use for blood group are slightly different. For blood group A, if the individual is homozygous, he would have capital I superscript capital A, capital I superscript capital A, if he's homozygous. If not, then it is capital I superscript capital A and a small I, showing recessive. On his blood, the protein antigen that he has is antigen A. And in his blood, he has an antibody for B. So in other words, if a person who is blood group A gets blood B, it could cause problems because the antibodies will fight and clot the blood. He can receive from A or O. B, IB, small I. Homozygous, heterozygous. Antigen, obviously B, antibody for A. He can receive or she can receive blood from B or O. AB, IB, it's heterozygous. It can only exist in that way. IB. Antigen, obviously, we got both, it will have both. Antibodies, because it got both antigens A and B, it cannot have an antibody because it's got A, it's got B. So no antibodies. What's good about that? It can receive blood from all because it doesn't have any antibodies. Blood group O, 2 small i's, antigen none, antibodies A and B, can receive blood only from O. While O can go to all, as you can see, O. All types of blood will get O, but O can only take O. Okay? Example, they say, show the cross between a mother who has blood type O and a father who has AB blood. Now, there's our spot to do this. Okay. So the parent, we're saying the mother has blood group, let me go back up, O, and the father has AB. Okay. So the phenotype father AB, mother blood group O. The genotype would be capital I A, capital I B. And the mother would be 2 small i's. Oops, 2 small i's. The gametes would be I A, I B, and I. Notice we separate the gametes. When fertilization takes place, we'll have IA. As a general rule, guys, I say your gametes that are on the left, keep them on the left, and the gametes on the right, put them on top. It's not necessary, you can switch them around, but just for your sanity and for your order, keep what's on the left, and keep what's on the right on the top. It becomes easier. So we take the one on the right, put it on top, I. And we do our cross. Notice the format is exactly the same. IA, IB, IB, Notice that all the children, possible children that can be born out of this wedlock are heterozygous. But none of them are AB. There's no chance, even though the father is blood group AB, there's no chance of them having a child that is blood group AB because the mother is O and O is recessive. And recessive can only show itself if it is in the homozygous condition. So each of them, you get one. Remember, how does this whole thing work? You're getting one gene from your father and one gene from your mother. This is what the square is telling you. These are the 2 genes your father can give you. These are the 2 genes that your mother can give you. So each child must have one of the small I and one what the father gives. Now this father is giving you one of 2 genes, with the A and with the B. Are you with me? That's what we can get there. Now, going back a little bit, somebody posted a question earlier that I'm confused about genotype and phenotype. Remember, genotype deals with the genes. You're either homozygous or heterozygous, or you show it with letters. For phenotype, tells you what is what we can call. When we do a blood test, we can't see whether the person is IAB or small I. We can either see whether they are A, whether they are B, AB, or O. So this is the phenotype of the individual. We can either see whether the flowers are red or white. That's the phenotype. Whether it's homozygous or heterozygous is the genetic makeup. So genotype, genetic makeup. And there we have that problem solved for you. Now we go back up a little bit to round up our theory here. Polygenic traits. Now, multiple alleles, there are more than 2 genes controlling this particular characteristic, but in one individual, only 2. Polygenic inheritance, individual heritable characters are found to be controlled by groups of several genes. So in one individual, you're getting more than just 2. You're getting more than that. That is polygenic inheritance. Each allele intensifies or diminishes a particular trait in the phenotype. That means it adds to the phenotype. How does it do that? It brings about a continuous or quantitative variation. What is a quantitative variation? It is different from what we said earlier, a discontinuous variation, where it's either red or yellow. It's either black or white, tall or short. It was an either-or situation. And Indy was talking about earlier when she said, what about different heights of people? And that's what we are talking about here. Look at this one here, seed color in wheat. It has AA, BB, CC, or AA, whatever, whichever way you're looking at it. We start with this one here is light. The next ones, they're all intermediate. And the last one is dark. So if an individual seed has this, it's dark. And if you take away that and you put anything in there, it contributes. It either adds or it diminishes a particular character. So the more capital letters, we're moving towards darkness of the seed, that is. And if we have less capital letters, we move to small letters, it diminishes. How does it diminish? It makes it lighter. Skin color in humans, by the way, is also controlled by polygenes. And height in humans is also controlled by polygenes. We don't either have tall humans or short. We're having short, shorter, taller, 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 and tallest. You can go right across the range. So it's a continuous, that means it's a gradual move from one to the other. It's not either this, tall or short. Okay? Lastly, a test cross is designed to reveal whether an organism that is dominant is homozygous or heterozygous. You'll agree when I say that the law of the cat and the mouse, when the cat is away, the mice will play. Obviously, for somebody to have the recessive characteristic, they must have homozygous. So when you look at the recessive condition, it'll always be homozygous. But when it's brown eyes, for example, in humans, which is dominant, we don't know whether the individual is homozygous or heterozygous. So we do a test cross with those individuals. We can't do that with humans, but we can do it with other organisms. What are we talking about? We can mate those organisms to see the offspring. If, for example, if red is dominant, and we make this flower red and red, and we only get red offspring coming out, that means that individual should be homozygous for that. If we get red and in between we get one white or few white, that means this individual had to be heterozygous. To get the white, the individual had to have the white gene to get the white into that flower. So that is what we are talking about when we come to a test cross. I'm hoping we all understand. On the page, guys, that's sure you're not going to see clear on the board itself, but on your page, on your notes, we've given you a format of all the possible genetic crosses: complete dominance, incomplete dominance, codominance, and sex-linked diseases. Use this as a recipe for every genetic problem that you solve. Go back to it and substitute the letters with other letters. I'm hoping you've been enjoying what we've done today. We're going to continue to next week on what we're doing. I hand you over to Ginny. Ginny.
Genie. I'll take it. Thank you so much,
Indy
guys.
the genie.
Indy the genie. I like. Guys, we've had such an amazing lesson. You guys have been posting on the page like champions. I want to say a big thank you to all of you guys. If you go and look on the page, please help each other out. Post if you know the answers, because sometimes you learn by helping everyone out. Aslam, everyone says that you were fantastic, that you rocked this lesson. You have been amazing. Grade 12 Mindsetters, you
guys.
are amazing. Thank you so much for joining in. You guys rock. See you same time, same place next week. Bye.